Hello children, welcome to Inside Engine Academy. In this video, we will discuss a lesson of class 10th is acids, bases and salts. Right? In this lesson, we discuss what are the various types of acids, bases, salts present and we use, and what are the different properties of acids, bases, salts, and how do we prepare acids, bases and salts, and what are the various properties, various types of acids, bases and salts. In this lesson we are going to discuss. Okay, before that, first let us see what are acids, bases and salts. In nature, we use various substances in our daily life. These substances based on nature are classified into three types of substances. One are acidic substances. Second, number two is basic substances. Third one are neutral substances. These are the three types of substances: acidic substances, basic substances and neutral substances. Let us see, for example, acidic substances. Hydrochloric acid. You might have heard this hydrochloric acid, which is one of the liquids, one of the juices produced by our stomach. Whenever we eat, our stomach used to release some kind of acids. Mainly it is dilute hydrochloric acid. This dilute hydrochloric acid generally digests the food in the stomach, like it any germs are present in the food that we intake are generally killed by this dilute hydrochloric acid. It is one of the gastric juices produced by the stomach. Let's say second, basic substances. For example, soaps. Soaps are the basic substances that we have. Let's say baking soda. We know baking soda. What is the common name, like chemical name of baking soda? Chemical name is sodium bicarbonate and its chemical formula is NaHCO3. This is one of the basic substance. Soaps are the basic substances that we generally see in nature. Okay, see, whenever we get acidity in the stomach caused by dilute HCl, and to treat it, baking soda solution is consumed. This is the base, this is the acid. What happened? Their effects are cancelled with each other when they are present, like same time. Thirdly, neutral substances. The very good neutral substance example is water that we take. Water is neither acidic nor basic. What about I'm talking this water? This water is if it is a distilled water, if it is a pure form of water. But you know, we use alkaline water generally, which is actually basic in nature, and lot of minerals are added. That mineral water we say it is not neutral. I'm talking about distilled water. Distilled water is a neutral substance in example. So in our everyday life, we use different substances which are acidic in nature, basic in nature, and also neutral substances we use, right? And what are these different substances, and why are some substances called acids? Why are some substances are bases? Why are some substances are neutral? What are salts and how the salts are produced from these acids and bases? We will discuss now. But before we discuss all those things, firstly, let us get some examples of acids, bases that we generally use. Let us see now examples of acids, bases and neutral substances. Let us see some of the acids that we see, like curd or sour milk, lemon water, vinegar, etc. And along with this, there are so many other things like tamarind juice, right? Apple juice. These all are generally contains acids in them. Next, let's see some common bases. Cucumber juice, bitter gourd, soap water. These are etc. basic substances. Next, water. If you can see water, common salt solution. Common salt is nothing but sodium chloride. Common salt I am writing NaCl solution. It is also a neutral substance. But among these various types of substances like acids, bases and salt, neutral solutions, how to identify them? Which one is acid, which one is base, which one is neutral? Okay, we use a substances called indicators. Substances called indicators to identify the nature of the substance, right? These different types of indicators are used in order to identify the substances, classify the substance into acids, bases, and neutral substances. And what are the different types of indicators that we have, how those indicators help us in identifying the nature of the substance, we will discuss now. Now let us see the indicator. What is an indicator used here? Indicator is a substance that is used to identify whether the given solution is acid, base, or neutral, right? There are, based on the source, there are different types of indicators are there. The indicators are mainly two types. One is natural indicators. Second one is synthetic indicators. Synthetic indicators are also called artificial indicators. Let us see what are these two different types of indicators. The first one is natural indicator. Natural indicators. So right, there are different types of natural indicators, and these natural indicators, is one of the natural indicators. Litmus. What is a litmus? Litmus is actually a type of lichen. A type of lichen. It is a plant species. This lichen, a liquid is extracted from lichen. Extracted from this lichen is called a litmus solution, and this litmus solution available, it is actually exist in purple color. It is a purple colored substance, litmus solution, and this litmus solution, it is one of the natural indicators, as it is extracted from a plant species called litmus lichen. It is a natural indicator, and this natural indicator, the original color is purple, and this purple colored litmus is used to identify acids or bases. Let us see. First, there are, in litmus solution we can make into two different types. One is a alkaline litmus, alkaline litmus solution. Second one is a acidic litmus solution. To the purple color litmus solution, what? A dilute alkaline stands for dilute. It's actually a base. So a dilute base is added to litmus in order to make a alkaline litmus solution. This alkaline litmus solution actually exists in blue color. Exist in blue. Purple colored litmus, it turned to blue color, and this blue color litmus is also a what? Alkaline litmus solution. Whereas second type is acidic litmus solution. From the purple colored we can make acidic litmus solution, and into this acidic litmus, in order to make acidic litmus solution, to the original purple colored litmus solution, dilute acid is added. Dilute acid is added, and this dilute acid like litmus is in red color. It is a red color. So now we have two different litmus. One is a blue litmus. Blue litmus, it is a alkaline in nature actually. Next, second one is red litmus. This red litmus is acidic in nature. Okay. The purple colored litmus solution has changed to blue litmus and red litmus now. This blue litmus and red litmus solutions and now are used to identify the nature of the substance. Firstly, blue litmus. If blue litmus is added to acid, which is actually a alkaline substance, when this blue litmus is added to acid solution, a chemical change takes place. A chemical change in the structure of the litmus change place. Because of this chemical change, there is a change in the color of the solution, right? This blue litmus solution, alkaline litmus solution, when reacts with acid solution, it turns to red color. The solution turns to red color. So we identify whenever a blue litmus turn to red color, the substance that is used to identify is acid solution. Then coming to red litmus. Red litmus is actually acidic, right? Purple color litmus solution is made acidic. When it added to acid, the structure changes, so its color changes to red color from purple. And now this red color litmus is added to a basic solution. Its color changes to blue. The solution color changes to blue. In any solution, if a red litmus turns to blue color and the solution that is used is basic, so that we use this red litmus and blue litmus to identify the nature of the solutions. So we'll write now these three types of substances, how litmus is used to identify different substances and different color change in different natured solutions. A color change of litmus in various solutions. First, let us see blue litmus. When blue litmus added to acids, it changes its color to red. Blue litmus turns to red in acids. Blue litmus remains blue in bases, and blue litmus remains blue in neutral solutions. Neutral solutions cannot change the color of litmus. Whereas a red litmus, if we take color in acids, remains red color, red litmus remains red. Red litmus with bases turns to blue color, right? In neutral solutions, red litmus remains red. These are the different color changes that we see to identify the nature of the solution using the two types of indicators, that is two types of litmus. One is blue litmus and the second one is a red litmus, right? Now, this litmus is available in two forms. It is available in liquid form, and also in the form of a litmus paper. Available in the form of litmus paper. In liquid form, we have two types of liquids, two colored liquids. One is blue litmus, second one is red litmus. Solutions, red litmus and blue litmus solutions. In the form of paper also, it is available in two colors. One is a blue, second one is red. Blue litmus paper and red litmus paper. These litmus solution or litmus papers are used to identify the nature of the substance, right? Now, if a blue litmus paper, red litmus paper, we have already seen what changes in color is seen when this substance is added to acids. Now, I will ask you a question, right? You try answering this question. If changing the color, if a litmus solution or a litmus paper is dipped in acid or basic solution, there is a change in the color. Is that change a physical change or a chemical change? You pause the video and think of it. If you try to answer and you verify, if you get any answer, then resume the video and understand whether your guessing is correct or not. Let us say here, color change when indicator is added to a substance is a chemical change. As I told you, what happens is whenever this indicator is added to acid or basic solution, there is a change in the structure of each molecule of the indicator, and as a result of change in the structure, what happened? Its color also changes in its solution, in its medium. So that changing color in acidic or basic medium when indicators are added is an example of chemical change. Right? Now, based on this litmus color changer, let's have one important question, right? This question, listen the question first and try to answer yourself by pausing the video. If you can, okay, it's right. Otherwise, you verify your answer from this video. Okay. Now let us say you are given three solutions. Okay. Solution A, solution B, solution C. Three solutions are given, in which in these three solutions, one is acid, one is base, one is neutral. But which one is acid, which one is base, which one is neutral, we do not know. In that case, we are given, we have to identify these three solutions into acid, base or neutral. For identifying this, we are given litmus indicator. But out of these two available litmus, we are provided only one litmus. It can be blue litmus paper or red litmus paper, any one colored a bunch of papers. Let's say if a bunch of red litmus papers are provided, then how can we use this bunch of red litmus paper? I am repeating, it's not one litmus paper, it is a bunch of red litmus papers, and we are not provided blue litmus papers, only red litmus paper. So we can use any number of red litmus papers to identify this acid, base and neutral solutions out of these three solutions given. Okay, for that what we should do, using this, how can we identify the three different colored substances? Right? You pause the video and think, how can we achieve this? Okay, if you can't get the answer, then resume the video and listen to the answer. Now, we have three solutions ABC. Let us dip. Let's take red litmus. Okay. Let us dip each red litmus, one red litmus to each of these three solutions provided. Okay, this is a red litmus, this is red litmus. We are provided only red litmus, but any number of red papers can be used. When you dip a red litmus paper in a solution, we can see out of these three, one is acid, one is base, one is neutral. Okay, we can also know, right, color changes. What is it? We are given red litmus. In acids it remains red, in bases it turns to blue, in neutral solutions also it remains red. Out of this three solutions, in only one solution, the red litmus turns to blue color. What is that solution? If red litmus turns to blue, we can identify the solution is base. So out of this A, B, C, one is a base. For example, let us say this A is for a base. Okay, let's say B is not base. It is just a assumed letter, like a solution ABC, unknown solutions, and we could identify one solution A into base because this solution, like this solution, made this red litmus to blue color. Okay, so this red litmus to blue color, any solution it turns litmus, then that is identified to be base. And how about the remaining two solutions? In remaining two solutions, red remains red. There is no change in the color of the litmus paper. It is remains red. In that case, we cannot identify in this B and C which one is a acid and which one is a neutral. We cannot identify. Right? In that case, we are not, see, you don't get an idea, we can use a blue litmus paper, right? Or else, if this blue litmus paper you can dip in other, no, one litmus is only used once. It is not useful for reusing, right? In that case, what do we do? Now we can we identified one solution into base, and out of remaining two solutions, base was completed, remaining is one acid, one neutral. We have to identify using the same red litmus paper. Now what do I do? This solution A, which is basic in nature, this is I like transfer some amount of base into test tube B and some amount of base into test tube C. Is it clear? One is 1 into B, one base into some base into another solution C. Then what happens? Okay, we learned in the previous classes like what happens when acids are added to bases. Okay, acids are added to bases. Generally, you learned in 7th class. There is a reaction called neutralization. when acid solution is added to base, it neutralizes the acid and base, produces a salt solution and water. This process is called neutralization. We will discuss more examples of neutralization in our next topics. Okay, now what happens now? Acid when added to base, these two neutralize their properties each other, so that this is a base added to neutral and added to, like, one can be neutral, one is acid. Okay, let me write here. If base is added to acid, what happens? The resulted solution is neutral. If base is added to neutral substance, the resultant is basic only. Okay, because neutral it is affected by base when it is added, and the resultant solution is also basic. But when base is added to acid, these two neutralize their properties each other and resulting a neutral solution. And we also know in neutral solution red litmus remains red, whereas red litmus turns to blue color in basic solution. Okay, now what I do in this B and C, we have already some amount of base which was identified as a solution A. The solution A is added to solution B and C, so that in one solution, let's say after adding, let us use again red litmus paper to identify the nature of the solution, resulted solution, right? It is not pure now, it's resulted, it's a mixture, addition of solution A. After this, in these two, one is acid, one is neutral. One solution becomes neutral, like, because that is added to acid and become neutral. Another one is base. Now, for example, this solution, in this solution, if red turns to blue, this is the basic solution which is obtained by addition of this base to the neutral solution. So we can identify solution B is neutral. I repeat once again, it was earlier a neutral, but after adding base added to neutral and resulted solution is basic in nature, so that red litmus paper turns to blue color. Let us imagine now this, initially it was an acid. To this acid, when base is added, what happens? It neutralizes, a neutral solution is obtained. In that case, red remains red color, and we identify the original solution that was provided is acid solution. This way, using only blue color litmus papers, we can identify the three solutions into acids, bases and salts. Okay, this is one important question based on identification of type of substances by litmus paper. So the same the same question may be asked by, if you are provided blue litmus paper instead of red litmus. Okay, what happens when blue litmus only provided, one of these three solutions turns blue litmus into red color, that solution is acid, and that then acid is added to remaining two solutions, right? And we can identify the color change, so that we can identify which one is acid, base or neutral. Okay, this is a first type of natural indicator, and it's a how to identify different natures using the litmus solution. And second type of there are other natural indicators too, and second natural indicator is turmeric paper, and that turmeric paper we will discuss now. Now the second type of natural indicator is turmeric paper, right? This turmeric paper is also used to identify the nature of the solution given. How to make this indicator, turmeric paper? For that, we take some turmeric powder. So that turmeric powder is added to water, and then that turmeric paste is applied to a paper, then dried. This paper is our turmeric indicator paper, and this turmeric paper now is used to identify the nature of the solution given. How can we identify this turmeric paper with acids? If I write the turmeric paper in acids, we observe no change in color. For example, this is a turmeric paper, yellow color turmeric paper. If you add a few drops of any solution to it, if that solution has no change on the color of indicator. For example, the turmeric indicator, what is the color of turmeric? It's yellow, right? If this turmeric indicator remains yellow, then we understand it can be acid or neutral. With acid solutions and neutral solutions, we can see no change in the turmeric paper. If the added substance is base, if added a base, for example, soap water is taken, few drops of soap water is added to turmeric paper, and this turmeric paper is turned to reddish brown, then we can identify that the given substance is base. So turmeric paper, its color is not changed when acid or neutral solution is added, but it turns to reddish brown when soap water, like a basic solution, is added to it, so that we can identify using this turmeric paper the given solution is acid or base. And next one, red cabbage extract. Okay, in market red cabbage is also available. Generally available cabbage is in green color, but red color, it's not actually red color, it's a purple color cabbage is also available at vegetable markets. Using that red cabbage, we can make a acid base indicator. What is it? It's a red cabbage extract is an indicator. So for making this red cabbage extract, we need to do only a piece of red cabbage is taken and it is boiled in water until that water extract its color, like until a purple color extraction, the solution is obtained. After turning that water into purple color, we just filtered off all the red cabbage, and that red cabbage extract is now used as a acid base indicator. And this red cabbage indicators, it changes its color in different types of medium: acid solutions, basic solutions, and neutral solutions. In acid solution, this solution, like red cabbage juice, it turns to red from purple. What is its original color? Original color is purple. This purple red cabbage extractor is turned to red or pink in acid solutions, if it is added to acids. Then if it is added to basic substances, it turns to dark green. It turns to dark green or yellow. Green or yellow. It turns to green or yellow. We identify that the substance is base. And in neutral solutions, this red cabbage extract turns to blue color. So that by changing the color, red cabbage extract identify the nature of the solution that is used, that is taken. And lastly, one more natural indicator is hibiscus extract. This is also made in the same way that red cabbage extract is made, right? For this, we take petals of hibiscus flower, and these petals are added to boiling water, and what happened, that water changes its color. Okay, when this hibiscus extract changes its color to pink, which color it is? It actually turns to pink color. And this hibiscus petals then added to acids, okay, bases, and neutral solutions, right? And it changes its color. Hibiscus petal changes their color. In acid solutions, the color changes to very bright red, we can say bright red color. So the solution that changes hibiscus solution into like bright red color, that the substance is called acid, identified to be acid. And bases turns this is into dark green or black. In bases, hibiscus extract turns into dark green or almost black. Then in case of neutral, we see no change in color. So by changing the color, this hibiscus extract is able to like indicate the nature of the solution. So these are the natural indicators. You see, litmus is obtained from a plant species Turmeric paper obtained from turmeric, it is also a plant product. Red cabbage is a vegetable. Hibiscus is a flower. These all are natural substances and are acting as a indicator, acid-base indicators to identify the nature of solution given. Okay, so because these are natural substances and you acting like indicators, so these all are called natural indicators. They are helpers in identifying the nature of the solution. Along with these natural indicators, there are also few man-made indicators which are more effective, more reliable to identify nature of the solution. What are those artificial or man-made indicators, which are also called synthetic indicators? We will discuss now. Now we will see. So the two important synthetic indicators. Synthetic indicators are artificial indicators, okay, which is prepared from different other substances, which are also changes their color in different acid solutions, bases solutions in order to indicate their nature, right? Now let us see what is these indicators original color and how do they identify, how they indicate the nature of the substances by changing its color. Let us see first one, methyl orange. Methyl orange is a like complex structured indicator, right? And this complex structure indicator, when added to acids or bases, what happens generally, its structure is being changed. Due to change in structure, its color also changes. Changing its color, we identify the nature of the solution. So first methyl orange. What is its original color? We can see its original color from its name. We can identify it's methyl orange. The original color of this methyl orange indicator is orange, right? Then how does this methyl orange color changes in acidic solutions and in basic solutions, right? It changes. This methyl orange indicator in acids, in acidic solutions, it turns to red color, and in bases it turns to yellow color. Methyl orange, it turns to red, and in bases it turns to yellow color, right? Its original color is orange. Then let us see another synthetic indicator is phenolphthalein. Its original color is actually colorless. It's actually colorless indicator. This colorless indicator, when it is added to acids, there is no change in the color. It remains colorless in acidic solution, but in basic solution, when this phenolphthalein is added to base, it turns to pink. Phenolphthalein turns to pink color. So that by identifying the change in color, we can identify what is the nature of the solution that is used to test its nature, right? Methyl orange and phenolphthalein, these are the two more commonly used artificial or synthetic indicators, right? Now let us discuss another type of indicator. If for example the indicators that we seen till now, natural indicators or artificial indicators, they all changes their color when added to different natured solutions. By identifying the color change, we can identify the nature of the solution in which the indicator is added. Okay, and how about the people of blind or semi-blind? They cannot identify the color, change in color, in order to identify nature of solution. Okay, and to make them understand the nature of solution, we have another indicator, that indicator called olfactory indicator. Here I would like to tell you, olfactory, in a human brain we have a nerve, specially made nerve called olfactory nerve. This olfactory nerve connects the nose to the brain, so that this nerve gives different sensations of different odors, different smells, and to the brain to understand. Okay, so olfactory means something is odor. It's odor or simply we understand its smell. Okay, so here olfactory indicators are the acid base indicators. They are generally changes their odor and with different natured acids and bases. Whenever they change their odor, we can identify the substances into acids or bases. And one of the commonly used olfactory indicator is onion indicator. Why do we use onions? Because onions have that characteristic odor, that pungent smell that the onion has. Because of this, onion is used as the olfactory indicator. For preparation of this onion indicator, what we have to do? So we just have to take an onion, right? An onion. So chop it into pieces, and these chopped onions are taken into a cloth, a clean washed cloth is taken. All the chopped onion pieces are tied in the washed clean cloth, and overnight it is kept in a refrigerator, right? And next day morning the onion pieces are removed from the cloth, and that cloth, if you smell the cloth, you will see that onion smell, that pungent smell. And now a strip of that cloth can be used as a onion indicator. So for that, what do? We just take an acid solution. For example, this is an acid, let's say dilute hydrochloric acid. We dip a strip of onion indicator, right? Then after dip, we just take it out from the solution, and if you smell it, if this strip of onion cloth still smells like onion, then the given solution is acid, right, or neutral. If you take another strip of onion cloth, what is this? This is a strip of onion cloth. This onion cloth dip in a basic solution, any basic solution, and after dipping, if you just take it out, we have to dip it completely. Dip this paper, like strip of onion cloth, completely into the basic solution, and you just remove from the basic solution and you smell it. What happened is onion smell completely vanishes. If the smell of onion completely disappears, completely vanished, then we can identify that the solution used to dip the onion cloth is basic solution. So by changing odor, the cloth, the strip of onion cloth is also used as a indicator to identify acid and basic nature of the substances, and this is one of the olfactory indicators. And second one, we also use a vanilla essence as a olfactory indicator. This vanilla essence also has its characteristic odor. Same, when it is added to acids, we can see no change in smell or odor, but when it is added to bases, what happens? Its odor disappears, vanishes. From this we identify the substance taken is acid or base. These are the two different another type of indicators like olfactory indicators. So that we could discuss three different categories of indicators. One is natural indicator, second one is synthetic indicator, third type is olfactory indicators. First two types of indicators, natural indicators and synthetic indicators, changes their color with different nature of solutions, whereas olfactory indicators indicate the nature of the substance by changing its smell, changing its odor, such are called olfactory indicators. So this is the different identification of acids, bases and neutral solutions using various indicators. Okay, and in our next class, in next video, we will discuss acids, bases, salts one by one. Okay, what are the various properties of acids, what are the different types of acids, right? What are the different uses of the acids? Same way, types of bases, properties of bases, uses of bases we'll discuss in our upcoming videos. Okay, I hope you understand this video. Thank you for watching.